the last thing that we have to talk about, yes, we have finally reached the end of this chapter. So the final thing that we have to cover for this chapter is known as identifying evolutionary relationships. Now, what does it mean by an evolutionary relationship? It just means that in one of the previous videos, I told you that evolutionary relationship basically explains to us where new species comes from. For example, there is a particular type of mammal species that existed hundreds of thousands of years ago, or even up to 1,000,000 years ago. And through evolution, they formed these three species currently present in the world. So that is evolutionary relationship. It's just to show you how evolution gives rise to new species. Now, in my example here, I have 3 islands. Please do not memorize it. But on these 3 islands, there are 3 different species of animals living separately. Species A, species B, and species C on each of the islands. The reason why the scientists say that they are different species is perhaps because they look different and they don't recognize each other as potential partners, and they cannot interbreed with each other to produce fertile offsprings. But still, scientists want to know whether these 3 species actually came from a common ancestor. So the question over here is, because the common ancestor might not exist anymore because it has gone extinct. So the hypothesis or the assumption is there was a common ancestor, and that common ancestor evolved and speciated to become species A, B, and C respectively. So the important question here is, how do we prove that these 3 species came from a common ancestor? So one of the main ways that we do it is we compare the base sequences of DNA. We can compare 2 types of DNA base sequence. We can compare base sequence of nuclear DNA, or we can also compare the base sequences of mitochondrial DNA. Because remember, if you're a eukaryotes meaning to say you have a nucleus, okay, and membrane-bound organelles like mitochondria, that's chapter 1. So if you're a eukaryote, you will have DNA in 2 places. You'll have DNA in your nucleus. You'll also have DNA in the mitochondria. So which one would scientists use to compare between different species? Would scientists compare the different species nuclear DNA, or would it compare the mitochondrial DNA? It really depends on the situation. Both can be used. There are some advantages of comparing nuclear DNA, but there are also advantages of comparing mitochondrial DNA. Now, before we talk about which comparison is better, we do need to cover the inheritance of nuclear and mitochondrial DNA. So, for example, in humans, this is for humans, but it can also be applied to animals as well. Okay, and even plants, by the way. So the first thing we have to understand is, for the male parent and the female parent, yes, that's the nucleus. I know the nucleus needs to have 46 chromosomes in the diploid cell, but for the sake of simplicity, I'm just going to put 4 chromosomes in the nucleus for the male parent and 4 chromosomes in the nucleus of the female parent. And the green color ones, if you're colorblind, it's okay, I'm just circling that. That reflects one set of chromosome, and the orange ones, which I'm circling, represents another set of chromosomes. So this cell is a diploid cell because it has 2 sets of chromosomes. Now, of course, also the cell would have a mitochondrion, not just one mitochondrion, but they can have multiple mitochondria, but I'm just drawing out one for each cell. For example, the male parent has a blue color, just for the sake of representation, just to represent it. The color of the mitochondrion is blue, and that's the circular DNA. Okay, the circular DNA is represented by that light blue color. So it's inside that mitochondrion. I hope you can see that. And for the female parent, they also have a mitochondrion. But I'm going to represent the color of the mitochondrion and the color of the mitochondrial DNA. By the way, mitochondrial DNA can be written in a short form as mtDNA. So it's the same thing. I'm just representing the color of the mitochondrial DNA differently. This is to tell you that the mitochondrial DNA in the male parent and the mitochondrial DNA in the female parent are different. So is the nuclear DNA, but I didn't change the color for that. Anyway, so when for these organisms to reproduce, what needs to happen is they need to undergo meiosis, because meiosis produces gametes. Now, due to meiosis, processes like independent assortment and crossover, it would cause the cell from diploid to become haploid. We know that it halves the number of chromosomes, but look at the color of the chromosome. It's not just totally orange or totally green. It's a mixture of orange or green. That's due to the crossover that happened, because remember, crossover causes exchange in genetic information. But the interesting thing is the mitochondrial DNA does not change. The mitochondrial DNA from here and here is exactly the same. For the female parent as well, when they undergo meiosis to produce the gamete, the nuclear DNA will change, because from diploid it becomes haploid, and from crossover and independent assortment, the base sequences in the chromosomes will be slightly different in the nuclear DNA. But interestingly, the mitochondrial DNA for the female parent also does not change in the gamete. So for the male gamete, the mitochondrial DNA, they are exactly the same. For the female parent, the two mitochondrial DNA is also exactly the same. Now, why is this important? Because when fertilization happens, okay, this is important. You see, the offspring will receive one set of chromosome from the male parent, which is those two chromosomes right there as I'm circling, and it will receive one set of chromosome from the female parent as well, which I am circling over there, okay, as you can see that. But here's where it becomes interesting. The offspring will only receive mitochondrial DNA from the female parent. So the offspring receives nuclear DNA from both parents, but the offspring only receives mitochondrial DNA from the female parent, or from the mother only. You don't have to know the reason why, but if you just want me to explain it, it's because the female gamete is quite large. And for example, in sperm cell, when the sperm fuses with the egg, only the nucleus of the sperm enters the egg cell. The mitochondria of the sperm remains outside. So it's not included in fertilization. So the offspring will only receive mitochondrial DNA from the mother. So, for example, the nuclear DNA over here, I'm just highlighting it. As you can see that I'm highlighting over here, the nuclear DNA from the male, the mitochondrial DNA is passed to the offspring, all right, only from the mother, by the way. But for the nuclear DNA, as you can see here, from the male gamete from the parent, it goes over there, and from the female gamete of the female parent, it goes over there. All right, so why is this such a big deal? This is a big deal because when you compare the nuclear DNA of the offspring, the nuclear DNA of the offspring is different from the parents due to crossover, independent assortment, random fusion of gametes, mutation, and so on. But the mitochondrial DNA of the offspring will always be exactly identical to the mother's, unless mutation happens, because mitochondrial DNA does not undergo crossover, there's no independent assortment or whatever. So why is this such an interesting thing? Because you can use the mitochondrial DNA to compare inheritance between parents. For example, you have a family tree over here. Square represents male and circle represents female. Now, remember, the mitochondrial DNA is passed from mom to child. So I've highlighted that mother over there at the top. She will pass her mitochondrial DNA to her 4 offsprings, all 4 of them. So that means the daughter will receive it and the sons will also receive it. And here's the thing. Can the son pass down the mitochondrial DNA to the child? No, it's only passed from mom to child, not dad to child. So it stops there. But what about that daughter? Can they pass the mitochondrial DNA to the child or the offspring? Yes, they can. So it gets highlighted over there. The child receives it. So those who I have highlighted in yellow, right, they will all have exactly the same mitochondrial DNA sequence, unless mutation happens. But assuming mutation does not happen, mitochondrial DNA are identical for all these people. And we can use the mitochondrial DNA as comparison to see who our parents are. So for you as a person individually, your nuclear DNA will be a mixture of both your biological parents. However, your mitochondrial DNA is exactly the same as your biological mother, right? So in that situation, your mitochondrial DNA will not be exactly the same as your biological father. It's only from the mother, by the way. So I hope you understand that part. So now that we understand the inheritance of both, how can we actually compare how can we prove that these three species came from a common ancestor? Well, what you can do is, number one, you can compare the base sequence of nuclear DNA. Now, some students will say, hey, you just mentioned that nuclear DNA changes due to crossover and independent assortment and random fusion of gametes. That is true, but certain parts of the chromosomal DNA, you don't need to know this in detail, but certain parts of the chromosomal DNAs are protected from crossover. So those parts cannot easily change, and those parts are referred to as the gene. So for example, let's say species A, species B, and species C have chromosomes. You compare the genes that code for the same type of proteins. For example, let's say when you looked at the chromosome of species A, B, and C, you notice that even though they are different species, they all have a particular similar gene, which is gene X. And gene X codes for protein X. Now, some students will be like, oh, what? Can you give me an example in real life? One example in real life is the hemoglobin gene, the HBB gene. The HBB gene is not just present in humans to code for hemoglobin or the beta globin. It's also present in cats, it's present in dogs. So we can use that gene to compare the relatedness between species. So for example, let's just use gene X over here. So for gene X over here, what exactly happens is we identify the sequence, we sequence the gene. Now, when we sequence the gene here, I know that genes for the DNA should be double-stranded, but I'm just going to focus on one strand, by the way, so it makes it easier. So we have the sequence of the gene over here. What we can do is we can compare the base sequences for species A and species B, and species B and species C. We notice that they are quite highly similar, but between species A and species B, there are 5 differences, which I've highlighted. But between species B and species C, there are only 2 differences. This would imply that species A and B are less closely related because they have less similarities, but species B and species C are more closely related because they have more similarities or less differences. Some students will then ask, how do we know that they all came from the same species? We know that because if they did not arise from a common ancestor, how is it possible that they all have gene X simultaneously? The fact that they all have gene X, yes, the sequence is slightly different, but the fact that they all have gene X would imply that they arose from a common ancestor. So what this implies is long ago, there was a particular species that existed. That species diverged due to separation and they undergo speciation to become 2 different species. This existed maybe thousands of years ago. That triangle-shaped species would evolve to become species A, but that square ones will then diverge further to become species B and species C. So this implies that species B and species C would have a more recent common ancestor, but species A, B, and C share a less recent common ancestor. Now, this is by comparing nuclear DNA. What you can also do then is you can also compare mitochondrial DNA. Now, why would we want to compare mitochondrial DNA? There can be many reasons for that. One of the main reasons is the cell has hundreds of mitochondria, so mitochondrial DNA is easily obtained. Number 2, mitochondrial DNA does not undergo crossover or random assortment, so it has many parts of the DNA-based mitochondrial DNA sequence can be compared fairly, all right? And the third thing that makes mitochondrial DNA as a suitable molecule for comparison is the fact that mitochondrial DNA mutates at a fairly constant rate. This is referred to as the molecular clock hypothesis. Now, you don't need to go into the detail of this and you don't need to memorize the value, but on average, there is an assumption that the mitochondrial DNA will undergo a mutation about one base every 25,000 years. It doesn't mean that, oh, after 25,000 years, the mitochondrion will bloop, just, mutate once. And then another 25,000 years, bloop, it just mutates another time. No. The scientists assume that when they compare fossil records, they notice the fossil records can be, up to a few million years old. When they compare the mitochondrial DNA of fossils and current organisms that survive, the average rate of mutation of the mitochondrion seems to be like one base every 25,000 years. Now, why is that point important? Let's talk about it. So over here, what we have is, we have the species A, species B, and species C, and we are not comparing their nuclear DNA, we are comparing their mtDNA or mitochondrial DNA. Again, I'm not going to draw out the double-stranded part. I'm just going to draw a single-strand sequence to make it easier. Species A has ACCTCAG. Species B has TGGTGCG. And species C has TGTTGCG. So what we do is we will compare their sequences. For species A and species B, they only have two similarities. What are the two similarities, by the way? At the T and also maybe the G. Right? But for species B and species C, they have 6 similarities. So when you compare the nuclear DNA, species B and species C had more similarities, species A and species B had less similarities. And when you also compare the mitochondrial DNA, it corroborates and it supports your assumption that B and C are more closely related to each other. So in this case over here, B and C share a recent common ancestor, but B and C share a less recent common ancestor with A. Mitochondrial DNA itself is not proof enough to identify ancestry or evolutionary relationship. You need to compare it with fossil records. You need to compare it with nuclear DNA. You might also need to compare it with the shape of the organism. So it's not good enough to use mitochondrial DNA as the absolute evidence, or it's not good enough to just use nuclear DNA as the absolute evidence. That's why evolution is quite convoluted, because you have to consider a lot of factors to even come to a particular conclusion. But remember, I told you about that molecular clock hypothesis. One base changes every 25,000 years due to mutation. What might have happened is scientists might have found a fossil record, and a fossil record of this species was about, it had a mitochondrion. Yes, sometimes if the mitochondrion is intact, you can get the mitochondrial DNA. That species, they are extinct, by the way. That's just a fossil record, and that is TGATCAG. All right, based on radiometric dating, we can determine the age of the fossil. The age of the fossil was about 80,000 years old, let's say. Okay, 75,000 to 85,000 years old. What might have happened is 75,000 years ago, perhaps, there was a divergence. Remember, separation, either it's allopathic or sympatric. They evolved to become two different species, and for every 25,000 years, the base sequence changed. And we're just going to put the base sequence in red where it mutated. If you are unable to distinguish if you are red-green colorblind, I'm just putting an underline there where the mutation happened. Remember, so between 75,000 years to 50,000 years ago, 25,000 years has passed, one mutation has happened. And from that organism that existed, after 25,000 years, the base sequence changes again. I'm just putting an underline over there. And for this one also, I'm underlining it. Okay, good. And then after another 25,000 years has passed, it has reached the present. It has become ACCTCAG. And the one at the bottom here, it would have undergone, perhaps, again, assumptions, it may have undergone speciation again. So that means they would have separated either allopatrically or sympatrically, okay? And they would have mutated again differently. So I'm just putting an underline there, just underlining it where it happened as well. So in this case over here, that's how you form those three different species. So based on fossil records, based on mitochondrial DNA analysis, based on radiometric dating of that fossil, we can assume that Species A, B, and C shared a common ancestor 75,000 years ago, but from 75,000 years ago, a separation happened. Again, you don't need to memorize my examples, just to tell you what happened. And about 25,000 years ago, another separation happened to form species B and species C. That's why they share a recent common ancestor. With that being said, this is how we identify evolutionary relationships, but these are not the only ways we do it. But the main two ways that you should know is just to compare the base sequence of nuclear DNA or the base sequence of mitochondrial DNA. That's what we do. But if it's a bacterium do not have nuclear DNA, they don't have mitochondrial DNA. So you just compare the circular DNA of the bacteria. That's normal, that's fine. So with that being said, chapter 17 is officially done. It has been a very difficult chapter to explain, by the way. So I hope you understand this, and please go through your past paper and get the hang of how the questions are being asked.